Hi, everyone, and welcome back to NPP Meet the Author series. Um, I'm Brianna Chen, and I'm Special Projects Manager at NPP.
I'm Brianna George, and I am the 2026 NPP editorial intern. And today I am very pleased to introduce Dr. Nakul Raval, and we're going to be talking about his paper called Multiple Sources of Beta 2 Nicotinic Acetylcholine Receptor Binding Are Differentially Affected During Tobacco Smoking Abstinence as Revealed by Independent Component Analysis of 18F Flubatine PET Images. Dr. Nakul Raval is an associate research scientist in the Department of Psychiatry at Yale School of Medicine. His interdisciplinary research bridges radiochemistry, PET imaging, and neurobiology to develop and validate novel radioligands and imaging tools for studying human brain function, with a particular focus on substance use disorders and the molecular mechanisms underlying neuropsychiatric disease. By advancing translational PET-based methods to characterize in vivo neurochemical and synaptic alterations, his work aims to deepen understanding of brain disorders and inform novel diagnostic and therapeutic strategies. So thank you so much for joining us today, Dr. Ravel.
Thank you. It's my
Um,
pleasure.
so our first question, so our first question is, in this study, you applied pharmaco-independent component analysis to your PET imaging. Could you first briefly explain what this component analysis is in simple terms, and then what motivated you to use this approach to study nicotinic receptor subtypes, and what were some of your main observations?
Yeah, so pharmaco-independent component analysis, or PHICA, is how we call it. It's essentially a data-driven way to unmixed combined signals. So think about the PET scan like a microphone recording an entire orchestra. So the radio tracer picks up the sound of all the different types of instruments that are playing, and the PET signal is basically a different radio tracer subtypes that this radioligand is binding to. So essentially, PHICA acts like an audio filter that separates the violin from the cello from the drums, and it also picks up where exactly that sound is coming from. So it isolates distinct signals or sources of PET signal that are unique in some way. Specifically, what we are trying to pick up is spatial patterns. So how different are these subtypes being picked up? So our motivation comes actually on very basics of pharmacology. So how PET tracers work. So a PET tracer or a PET signal is the combination of the amount of receptors or the receptor pool that you are imaging over the affinity of the radioligand. So it's a combination of the amount of receptors and the affinity of the radioligand. So if a radio tracer has distinct affinities for different subtypes, it can also be a bad thing, but in case where it's a good thing, which is in this case, with tracers like Flubatine. In general, it's a fantastic radio tracer which images all beta 2 nicotinic subtypes. But if you want to now separate these beta 2 different beta 2 subtypes into something like alpha 4, alpha 6, or alpha 3 nicotinic receptors, you can use this data-driven strategy to mathematically separate the signal instead of seeing the whole brain as one uniform receptor pool. So it's basically a data-driven strategy to do that. So the main observations that we found in this is that we found three highly reproducible independent components, or these components basically which are spatially distinct from each other, and we were able to replicate these different components in different data sets that we had recorded at the Yale PET Center. So the first two components that we find are mainly thalamic and the midbrain. So this is the main region where nicotinic receptor subtypes live. However, we found a very distinct cerebellum component, which we thought was really interesting. This was the alpha 3 beta 2 like component. So we're not completely sure that it is this subtype that we are imaging, but we were able to corroborate our in vivo findings using in vitro autoradiography. So that's exactly my specialty. I'm somewhere in the middle of translation PET imaging, where I have expertise in vitro studies as well as in vivo studies. And from these three components, what we found is that the thalamic components, both the thalamic and the midbrain components, were significantly lower in abstinent smokers. So these smokers were abstinent for about 7 to 14 days, and all these components were lower. And we know this from previous studies that it often is lower. However, this cerebellar component, which I was talking about, was significantly higher. So it was an opposite direction where it was going, and hence we thought this was a very interesting and novel finding.
I really love your analogy of PHICA as an orchestra. Do
Yeah.
you think that this PHICA framework could be extended to other receptor systems where subtype selectivity is limited? And are there other particular tracers or neurotransmitter systems where you think this approach would be especially powerful?
Yeah, absolutely. I think the potential is massive here. We actually built this framework on a previous work from our own group that successfully separated dopamine D2 and D3 receptors from a tracer called PHNO. So we know that our mathematical foundation was solid, and hence we kind of dive deep into one of this. So to answer your question, yes, there is potential on other work, and we are also pursuing this other tracer PHNO and going deeper into it. However, I know and for a fact that there are multiple other radio tracers where you can do this. One of the works that I'm really interested in is psychedelic research, and one of the perfect candidates for this is a radio tracer called SIMBI 36, which was developed by the group that I did my PhD in Copenhagen. It has affinity for both 5-HT2A and 5-HT2C. And because these receptor subtypes are differentially expressed in the subcortical region as well as the cortical region, I think ICA can theoretically unmix this signal, which will allow us to study a 2A as well as 2C dynamics simultaneously in human psychedelic research. And that's something which I do want to pursue in the future. Currently, I'm working on a grant which is funded by the NIH, specifically NIAAA. That's the alcohol and alcohol research part of NIH, and I'm trying to develop a gluN2B radioligand, which is an NMDA subtype. However, we've now found that we have significant off-target binding. So this would have automatically killed this radio tracer because of this off-target binding. But because we know that there is potential in using a mathematically data-driven strategy to separate out the signal, we can potentially save this radio tracer, and it makes it a boon as well in a way that if we know what this off-target binding is, we can separate the off-target binding from the gluN2B signal to give us two images from one single scan.
Amazing. That actually leads me directly into my next question. So, from a clinical perspective, how do you see some of this work informing treatment strategies for nicotine use disorder or cognitive dysfunction during abstinence? I know some of that was mentioned in the paper as well. And what excites you most about this translational potential?
Yeah, I mean there is of course the whole clinical standpoint. We as imaging scientists, we often care about how is this going to get translated into a clinical standpoint. And I think the most crucial takeaway is that chronic smoking doesn't just turn the brain's nicotinic system upside down, but it's a differential system that it's changing. So one of the receptors is probably upregulated while the other receptor is downregulated. So for instance, in our exploratory data that we were looking at, we found that the alpha 3 beta 2 signal was correlated with how much smoking was done in these abstinent smokers. And this kind of could reflect on why people have what they often call the brain fog when they are trying to quit. So we do know this, that alpha 3 beta 2 receptors are not hit directly by nicotine, so they don't have such a strong affinity for nicotine, but we know that these receptors are changing. So it directly takes us into this cognitive dysfunction question that you asked us, because it's highly relevant. Like alpha 3 beta 2 has been shown to be an important subtype when it comes to cognitive deficits. So people who have cognitive deficits often have lower alpha 3 beta 2 in their brains. And if that is what we are looking at, we can potentially help them quit smoking in some way or the other if we can target these specific subtypes. And to answer your last point, what excites me is the translational potential. Like it's how much we can go from in vivo data back into in vitro data and back into in vivo data. This whole cycle is what drives me as a scientist, to allow me to go back into the lab and corroborate my my in vivo results is something which I would love to keep doing. In the end, we are trying to target a very complex system. The cholinergic system is one of the systems which has multiple number of subtypes. So if in the future we are able to pinpoint to one of the subtypes which can help people quit smoking, which is, and we know is a bad habit, it could really be something for the future.
Amazing. This is some really great work. And thank you so much, Dr. Raval, for taking the time to speak with us today. And if you haven't already, please check out the article. It's going to be linked in the video description below. And as always, feel free to comment or reach out on social media. We're both on Blue Sky and X if you have any questions.
Thank you.